So good morning, good afternoon, good evening, and Happy New Year. Welcome. We are Irina Rogozina and Denis Didier Rousseau on behalf of Andrzej Volker, who couldn't make it. Today's convener of this webinar, the seventh in a series organized by Climate of the Past on one of the journal's topic. This series is part of the journal's celebration of its 20th anniversary of successful leadership. The webinar will last for 1 hour with two presentations of 25 minutes and conclude with a half an hour audience Q&A to be asked only on the Q&A box at the bottom of your screen. All webinars are recorded and uploaded to the EGU YouTube channel, thanks to EGU office. Today's topic is polar climate history. The polar regions play a critical role in the global climate system and are critical for understanding ice sheet dynamics and sea level changes. Currently, the continental ice sheets on Antarctica and Greenland are losing mass, and sea ice concentration in the Arctic Ocean are declining and the ocean waters warming. This session will explore the polar climate history with views from both poles and perspectives from the distant past in the Paleogene to more recent changes during the last glacial cycle and Holocene. To address this topic, we have selected two guest speakers, Peter Bijl from Utrecht University and Eike Zimmerman from the Geological Survey of Denmark and Greenland.
All right, so thanks a lot for the organizers to invite me to give a 30-minute webinar on the long-term history of the Antarctic Circumpolar Current. Yeah, I'm Peter Bijl. I'm an associate professor at Earth Sciences at Utrecht University. I was academically born and raised in Utrecht, and when they realized they couldn't get rid of me, they gave me a permanent position. So I'm here, and I'm part of a great team of mostly palynologists in this department, and I will mostly presenting results from PhDs and postdocs in my research group that I was happy enough to supervise. But also you will see this whole string of names, and those are all master students and bachelor students that did research projects with me. So I will be presenting their results as well. So let me see, I need to see how this works. So if we talk about the oceanography of the Southern Ocean, it's actually a complex interplay of a lot of different processes. So the Southern Ocean today is associated with very strong latitudinal gradients in pretty much everything you can think of, from ecosystems to temperature, nutrients, salinity, and also obviously the presence of sea ice. But it also has a complex deep circulation underneath, with regions of upwelling and intermediate water formation, bottom water formation close to Antarctica, of course. And all these different features are not gradually changing latitudinally, but actually are separated by sharp fronts that separate these oceans in different ocean zones. And it's best illustrated here in the bottom. I have to turn this plot around to go from Australia to Antarctica. You have this gradual decrease in sea surface temperature, but it actually happens in these strong shifts, and those are the fronts that separate different water masses. And so these fronts actually determine for a large part local weather controls. Different swings of these fronts determine what kind of weather Australia gets, for instance, but also direct precipitation regimes, and are therefore very important components of the Southern Hemisphere climate. And plus, they basically represent the latitudinal distribution of heat also across the Southern Hemisphere, including the heat that is eventually reaching Antarctica and supplies its precipitation and determines its temperature. And last but not least, or also obviously one prominent feature is the strongest ocean current on our planet, the Antarctic Circumpolar Current that circulates Antarctica mostly via these frontal systems. And yeah, that's also a very important feature in the Southern Ocean oceanography. And last but not least, it's the Southern Ocean is really important in ocean carbon exchange, because depending on where you're on the Southern Ocean, you have regions of that is a strong carbon sink or a strong carbon source of CO2. And so, yeah, I think this sums up the importance of the Southern Ocean in the climate system in the Southern Hemisphere, but also globally with global connections through the deep ocean circulation. And so, if we want to know how this system developed over geologic timescales, obviously it comes into play what climate did in the past, but also what the tectonic configuration was. And so, if we talk about climate of the past 65 million years, the community is pretty set on assigning, and I think rightfully so, a very dominant role on atmospheric CO2 concentrations that have shaped basically climate over the past 65 million years. And it is nicely summarized in this overview figure, where you see on top are sort of best guess atmospheric CO2 reconstructions over the past 65 million years. And in gray, a representation of global mean surface temperature, which is derived from the benthic foraminiferal oxygen isotopes with some assumptions. And you see the consistency between the trends in the two. So particularly high atmospheric CO2 levels during the early Eocene climatic optimum, and also high global mean surface temperature. And then from about 50 million years onwards, you see this gradual decline in CO2 and temperature. And then it is thought that CO2 crossed a certain threshold, so decreased below a certain threshold, that allowed the onset of the cryosphere system in polar regions. So first of all, at 34 million years ago on Antarctica, and then later in the mid-Miocene on the northern hemisphere. And that also, this whole concept also assigns a critical role to CO2 in being responsible for the onset of this cryosphere particularly on Antarctica. So yeah, CO2 is seen as the critical factor that triggered the Antarctic glaciation. But before that insight came to be, it was very long thought that actually the changing tectonic configurations played a part in shaping these climate trends. And so you can see in the Southern Ocean how fast the tectonic changes were in this tectonic reconstruction, with the quite astonishingly rapid northward movement of Australia, and also the separation between South America and Antarctica. And these two features basically gave way to a latitudinal belt around Antarctica that is unobstructed by land. And particularly this feature that is seen as the most important feature that allowed the Antarctic circumpolar current to develop. And the question now is exactly if these gateways that opened and that allowed for the Antarctic circumpolar current, if they did not really cause a glaciation because CO2 did, then what was the role of this tectonic change in shaping, first of all, the oceanography around Antarctica, and then what was the role of that in shaping the onset of Antarctic glaciation? Because it is actually quite coincidental that the opening of the Tasmanian Gateway, for instance, occurs exactly at 34 million years ago. So if not really being the main trigger of Antarctic glaciation, then what was its role? So those are the questions that I would like to address in this webinar. So what were the major steps in the development of the Antarctic circumpolar current and the associated oceanographic features that we see in the Southern Ocean? How do these actually link to developments in the Antarctic ice sheet? And what were the consequences of this evolution for the evolution of latitudinal temperature gradients and fronts in the Southern Ocean? And so for that, I will be presenting results from a series of proxies that we applied. I will introduce these proxies, obviously, throughout my lecture, but this all obviously begins with the availability of sedimentary records that capture this history of the Antarctic circumpolar current. And for this, obviously, I cannot avoid a big shout out to the International Ocean Drilling Program and its predecessors, who have provided this widespread of sediment sections that we can still use even with new proxies and new insights, because these cores are curated so well in the system. And so yeah, we cannot forget to think about how much more we know of past climate because of this program, and I have to say that also right here. And so, yeah, because proxies are constantly in development, it's actually really important to be able to go back to these cores and apply new proxies to these sediment cores, and that's exactly what I've done a lot in my career. So next to proxies, I will also be showing some key simulations of ocean models that are very useful in, first of all, comparing the proxy results to, but also extrapolating that to the entire Southern Ocean and providing sort of a physical oceanographic basis to the interpretations that we do. So my pet proxy, obviously everyone has a pet proxy, but this one is where I'm most expert of is dinoflagellate cyst assemblages. They are the cysts of unicellular algae that live in the surface waters. They have all kinds of different morphologies. There's a whole taxonomy associated to it. But we see in the Southern Ocean, I will show you later a result of that, that they are strongly associated to specific ocean zones as different species. And so if oceanography changes, we can see that in the dinoflagellate cyst assemblages that are preserved in sediments in the Southern Ocean. And that gives us actually a pretty sensitive proxy for temperature, nutrients, upwelling regions. We can identify whether there was sea ice, yes or no, and we can also identify salinity changes throughout the Southern Ocean. And we do that in two ways. So first of all, we have now compiled as a community of dinoflagellate cyst enthusiasts a compilation of surface sediment samples globally actually, but here you see the compilation for the Southern Hemisphere, and that now consists of about 650 surface sediment samples where we analyze the dinoflagellate cyst assemblage from. And also what we did in this particular study is look at some statistical analysis that cluster these sediment samples based on their assemblage, unconstrained by geography, yeah, based on their dinoflagellate cyst assemblage. And it came back with sort of 9 clusters, and samples in this cluster have basically a similar dinoflagellate cyst assemblage and are all dominated by one particular species. So you see that those sort of the average pie chart composition of that particular cluster in the back, and the dominant species of that as well. So what you notice in this map is that we color-coded these clusters, and you see that the clusters are actually very well banded latitudinally. So from dark blue, really close to Antarctica, to sort of a purple zone a little bit further away from Antarctica, going to orange and yellow, to the brightest yellow at the equator, north of Australia, for instance. And this gives us, and if you look at the ecological affinity then of these clusters, you can see that it actually has quite some tight and nice ranges and niches. So the advantage also that many of these species that you see on the right-hand side that characterize these clusters have actually long ranges of at least going back into the Neogene and some even towards the Eocene and Oligocene boundary. So that provides us also with a way to interpret ocean conditions really far back in time using this modern analog principle. And this is maybe a little bit complicated, but I've color-coded this for you. So these are basically the ocean zones that we distinguish based on the dinoflagellate cyst assemblages. And I will use this color coding throughout my presentation to guide you a little bit through the maze of species and assemblage plots. If we go further back in time to the Eocene, for instance, to investigate what the oceanography looked like prior to the Antarctic Circumpolar Current and prior to the existence of these species, we cannot use this modern analog technique anymore. But luckily, how do you say that Providence has provided us with another tool, namely that prior to the EOT, dinoflagellate assemblages in the Southern Ocean were characterized by this specific endemic community. They were called transantarctic flora. It was already recognized in the 80s. And this endemic dinocyst community occurs basically only in the Southern Hemisphere high latitudes. And already during my PhD, I sort of mapped out the distribution, the geographic distribution of the endemic community throughout the Southern Ocean. And you could see that their dominance is confined to the Antarctic margin, but also to the Southwest Pacific and the Southwest Atlantic Ocean. And it nicely coincided with, if you overlay sort of the modeled oceanography in the Eocene over this distribution that is shown in these arrows. So those are the outcome of model simulations of oceanography, if you close these gateways, then you see that there's a nice consistency between the two. So the regions where endemic dinoflagellated cysts dominate are regions that have Antarctic-derived ocean currents flowing northward, basically the western boundary currents of those subpolar gyres that now no longer exist because the ACC runs through. And the regions where those endemic dinoflagellated cysts are absent are the places where basically subtropical, like warm currents are captured. For instance, South Australia and the Australian Antarctic Gulf. And so that makes this dinoflagellated cyst distribution a pretty good confirmation of the ocean of the modeled ocean circulation prior to gateway opening. So just as we have in the northern hemisphere, the southern hemisphere with closed gateways has these strong subpolar gyres that circulate in the southern hemisphere high latitudes. And yeah, they effectively bring mid-latitude water, sort of temperate water, towards Antarctica. That is what they basically do. So another proxy that we use in our work here in Utrecht, also an organic proxy. Yeah? So the reason why we use them together is because they have the same preservation potential, is these biomarkers paleothermometry proxies. So I won't go into much detail about it, but we basically apply two temperature proxies based on membrane spanning lipids of two different organisms. And the molecular structure of these membrane spanning lipids is adjusted based on the temperature where they live in. So we can measure the relative abundance of these components, and also through modern core top calibrations, we can reconstruct temperature back in time. And so it's the combination of sort of the full oceanography from the dinoflagellates and specific absolute paleothermometry from the biomarkers that is used as proxy to reconstruct what the ocean looked back in time. In terms of models, the only thing I want to say here about it in terms of methodology is to stress the importance of high enough resolution models. And this is obviously a pain, because the higher resolution the models are, the longer they take to run and the more complex they are. But here I want to show briefly a clear reason why this is so important. You see on the top a model that is run in a 1 × 1 degree resolution, and in the bottom a model that is run in 100 times higher horizontal resolution. And you see three things basically happening. So the oceanography that is represented in the model is obviously more complex. The ocean currents are more confined, and you see representation of eddies indeed in this model. So it's actually more chaotic, but it's also more realistic in that way. And the final thing that you see is that actually you see much more representation of the bathymetry in the surface ocean flow. And this is really important because it is indeed more realistic. So that is also what happens in reality. It also puts the ball back to accurate paleogeographic and paleobathymetric reconstructions, because it really matters a lot if you're sitting east, west, north, or south of a bathymetric obstruction with your proxy site if you compare it to your model. So these are really important features. And just to show you a zoom in of this model, the high resolution model around Tasmania, you see the complexity of small colder currents and a big East Australian current that flows all the way deep into the Southern Ocean here. And that's a feature that was not really that well visible in the lower resolution records. And it does explain some of the features we see in the proxy data here close to New Zealand, the warmth that they represent. So shout out for high resolution modeling. Okay, so what we did specifically for the EOT, if we think about the oceanographic consequences of these gateway openings that were seen so important, right, for the development of the ACC, I will present the work of Isabel Sauermil, who did this work as part of her PhD at UTAS in Tasmania. I was happy to be part of that. She basically simulated also with a high resolution ocean-only model the oceanography of the Southern Ocean in a couple of configurations of the Tasmanian Gateway and Drake Passage. So you see these depth configurations here with the Tasmanian Gateway 300 meters, 600 meters, 1500 meters, and Drake Passage also at various depths. And we're going to compare these simulations to see what the consequence is of deepening of the gateway to oceanography and the distribution of heat. So you see here in the top panel, on the left-hand side, you see the ocean flow field, and on the right-hand side, you see the temperature in the simulations. And on the left-hand side panel with the flow field, you see that in this configuration, the Tasmanian gateway is shallow, it's only 300 meters, and Drake Passage is 1,000 meters. And you see here that the Southern Ocean features this strong subpolar gyre, and you also see the subtropical gyres in orange. And also the Atlantic Indian system has a strong subpolar gyre. So even though Drake Passage is deeply open, there's very little exchange between those two gyres. You see that here in Sverdrup and the arrows point to the direction of the ocean flow. So both gateways have very little connection, even though Drake Passage is deep. So now what happens if we keep Drake Passage deep and we start to open Tasmanian Gateway? What we effectively see is the destruction of both of these subpolar gyres. So the Pacific gyre is reduced in strength, but particularly also the Indian Ocean gyre, even though nothing is happening with Drake Passage. And you see that the ocean flow, right, eastwards is obviously the proto-ACC, the circumpolar flow. It's and I don't know if you know these numbers, but this is still 100 to 120 Sverdrup less than what the ACC does today. So there is circumpolar flow through those gateways, but it's nowhere near as strong as the modern day ACC. But you do see that it doesn't matter which gateway you deepen. You need to deepen the second of the two gateways, and only then you start to see some oceanographic consequences. So it doesn't matter if you have a sediment record here in the South Atlantic and you see a change that you interpret as a deepening of the gateway. It doesn't matter that you are close to Drake Passage, that it must be then Drake Passage that changes. It can also be the Tasmanian gateway. That's how well these systems communicate and how much they depend on the two gateways being deep. What you also see in terms of temperature is that the deepening of the gateways do have a consequence in the latitudinal sort of distribution of heat and the supply of heat of basically mid-latitude warmth towards Antarctica. So that was also a feature that we looked into, and I think it's an important change that the gateway changes could induce on Antarctica. They might not be big enough to really cause the Antarctic glaciation. After all, it was very well established that gateways don't trigger glaciations, but they may have been important in setting this threshold value of CO2 for Antarctic glaciation, making it possible to initiate a glaciation at higher CO2 levels than when the gateways were closed. So as a secondary accessory forcing factor, they might have played a role in this. So to conclude this EUT part, we see an onset of Antarctic glaciation, which is simultaneous to the first deep ocean connection between Tasmania and Antarctica. And we see that CO2 decline across a critical threshold is seen as the primary trigger of EUT glaciation. And the modeling suggests that there's no gateway change required to trigger this glaciation. However, so this idea of having a threshold of Antarctic glaciation means that I think there's all kinds of other boundary conditions that set this threshold, making this threshold higher or lower depending on the local geographic or climatic background conditions. And so, and I think that is what the gateways might have done during EOT. So, I need to skip a few things because I think I'm already. How much time do I still have?
5 minutes.
Oh, okay.
Make it.
And so, if we think about the Oligocene, I think it's characterized by this time interval where the Antarctic ice sheet was really variable in size. And I think it's important to think about what the role was of the developing Antarctic circumpolar flow around Antarctica that might deliver an explanation to the dynamics of the Antarctic ice sheet in the Oligocene. And so for that, we can look at the record of ocean changes, of ocean conditions close to Antarctica. And we do that by looking at the record site 1356 here close to Antarctica, where we have this nice dinoflagellate cyst record. It's a very long record of Oligocene sediments. Now I'm going to skip a few slides. So when we look at the dinoflagellate cyst there, we see indeed that at the Eocene and Oligocene transition, we see this blob of sea ice associated dinoflagellate cysts that occur today really close to the sea ice edge, but that disappears in the rest of the Oligocene and only returns at the mid-Miocene climatic transition. So what we see besides that is these light blue species which occur basically between the subantarctic front and the Antarctic polar front. And also occasionally we see the abundance of species that today occur close to the subtropical front. And that was really a surprise to us, that apparently the ocean conditions close to Antarctica in the Oligocene, even though there was an ice sheet on the continent, allowed for the presence of these warm temperate species that today live close to the subtropical front in waters that are between 10 and 15 degrees temperature. So if we look at the biomarker record of that site, that's here in red and blue, we were able to distinguish glacial maximum and interglacial maximum samples. So you also see this sort of 15 degrees. I think newer calibrations would bring this down a little bit, but around 15 degrees temperature for the Oligocene close to the Antarctic margin, which is warm if you think of it. But if you look at the dinoflagellate cyst images, it makes actually sense. Those two proxy data come really close to each other. So it shows you a completely different kind of oceanography in the Southern Ocean, right? Even though we have an ACC, we have some circumpolar flow, the oceanography must have been completely different. And I think to understand this kind of temperature record, you have to put it into context of other records in sort of a latitudinal transect. And that is what we did. So we generated records also from site 274 here in close to the Antarctic margin, but slightly further offshore. And from here, Tasmania. So this is a paleogeographic configuration that has Australia obviously much closer because it was the Oligocene. And we have these sites here to put the record from 1356 into a better latitudinal context. You see the color coding of the dots here, and that represents the color coding of these temperature records here. And you see a latitudinal temperature gradient through the Southern Ocean, basically, although it was narrower at that time, of about 5 to 6 degrees. And that is indeed a lot less than today, obviously. Yeah? So that shows us that the latitudinal temperature gradients, the strong gradients we know of today, were nowhere near as developed as in the Oligocene as they are today. Okay, then I want to say a little bit about, as a final thing about the Neogene, because we still don't have a modern ACC yet, right? So we had great fun with this site 1168, where we generated a lot of data from. This is a big shout out to focus on one site and generate high resolution data sets, because it's really worthwhile getting the long-term context, but also the amplitude of variability. So we have a nice bulk isotope record that ties the record to the xenogrid on orbital timescales. And we have a high resolution sea surface temperature record from two proxies from this site that shows the long-term evolution of the temperature of Tasmania while it moves northward. And what is particularly prominent in this record is this Neogene cooling, right? From the mid-Miocene to the modern, we have this strong cooling at this site. Okay, so from the dinoflagellate cyst, we know a little bit what kind of oceanography is there. I won't go into detail because of the lack of time, but we know very well where the subtropical front was relative to the site in this record. It was always really close. So while Australia moved northward, it basically dragged the subtropical front along with it northwards. So if we look at again the compilation of these temperatures in the latitudinal transect, we see obviously warm temperatures from the Western Pacific warm pool and a little bit of cooling, strong cooling in all these sites that come from the subtropical front area. and very little cooling in the Ross Sea. And that's because the Ross Sea was always close to land ice and that the ocean water was feeling that. But if you think now about latitudinal temperature gradients and particularly what cooling usually does to cooling in the poles with polar amplification, some interesting features appear here. So if you think this as the equator, equatorial temperatures cooled a little bit, and the steeper cooling here in the mid-latitudes, that's obviously polar amplification, right? So the subtropical gyre has amplified cooling towards its southern end. But if you look at the latitudinal temperature gradient from the mid-latitudes, from the subtropical front to Antarctica, you see something else. It has actually the strongest gradient in the Miocene and a weaker gradient in modern times. Yeah, so there's a lot more cooling in the subtropical front area than close to Antarctica. And I think this is an interesting feature, and it goes a little bit against the overall theory of polar amplification of cooling, but it also does something to the dynamics of the Southern Ocean if you have a sea surface temperature gradient that is as big as the one in the middle Miocene. And it's still difficult for me to grasp what the consequences of this is, but I think it was a really interesting observation that we should take into account when we interpret these kind of Miocene climates, particularly as analog of the future. I think there's a lot in the Miocene that is not analogous to the future. So, and if you ask this guy here, Evangelinos, he was part of Carlota's group and also collaborated with Tina van der Vliet at Imperial College. Based on neodymium isotopes, the first time we see an ACC with modern day strength is between 10 and 7 million years ago. And I think that's now where the state of the art is that the age of the first strong ACC becomes younger and younger if we move through time. And it's up to us to decipher what we would see in the temperature trends if this was really true. So that was it. And I thank you very much for your patience. I hope I wasn't too far over time.
No worries. It was a great talk. Thank you so much. Oh, sorry, my video is off. Yes, thank you, Peter. We'll come back to the questions that will be asked a bit later after the next presentation. Yeah, Heike?
All right. So I want to take this moment and want to say a big thank you to the organizers of the webinar for inviting me, and it's really an honor to speak here today. And now I want to take you to the other side of the globe, to the Arctic. And in this beautiful picture that you can see here from the book Plankton Chronicles, you can see a lot of different species and body types, and some of them you might even recognize. There are some diatoms and dinoflagellates that can be preserved over relatively long time scales as microfossils, but there are also a lot of different species there, such as crustaceans and worms and jellyfish, that we typically do not find in the sediment record, but we find them as DNA. And I guess most of you are aware that the climate of our planet was quite dynamic in the past. And here you can see a global temperature reconstruction over the past 60 million years. On the left side of this graph, we can see that there has been a dramatic cooling from the Eocene to the Pleistocene. And as Peter already said, this led to the onset of the southern and northern hemispheric ice sheets in the past. If we look more on the right side of this graph, we can see the temperature projections for 3 different carbon emission scenarios. And if we look here at a more intermediate scenario, such as the RCP 4.5, we already see that we're catapulting ourselves back into the mid-Pliocene, so around 3 million years ago. But for example, the last interglacial also reached temperatures that were quite comparable, and so the last interglacial that ranged from about 130,000 years to 15,000 years ago could also be an analog for some parts of the future. And if we look at the mid-Holocene, especially the mid-Holocene thermal maximum, we know that temperatures were also higher than they are today, so they could be an analog so this could be an analog for the near future. What we see from this is that it is critical that we study the past to better understand what may happen to the biodiversity in the future. Marine sediments are quite complex. Marine ecosystems, I mean, are quite complex. There are a lot of processes that are summarized in this figure, and there are different food webs, and the processes and the food webs or the species that were living under the conditions the processes created, they leave fingerprints in the sediments. So we can use sediments that are natural archives and reconstruct the past. Today I want to introduce to you one specific and relatively new proxy called sedimentary ancient DNA. Ancient DNA is giving us a lot of new opportunities, because if you look at this picture, you see that there are a lot of different species in the food web, and all of these species contain DNA. And the DNA can settle through the water column to the seafloor, where it can bind to mineral particles, get buried, and thereby also preserved over time. So DNA is an important new tool that we can use to better understand the long-term responses of biota to past climate. But we don't use sedimentary ancient DNA all by itself. We often add it to microfossil records or biomarker records. And now I want to give an overview over the strengths and weaknesses of the different proxies that we're using and the added value that I think sedimentary ancient DNA can have. So if we start with microfossils, they can be preserved over relatively long timescales, so in the range of tens of millions of years. There are large data sets available, and it's potentially quantitative. However, there's no genotype information available. It's quite labor intensive, and we need expert taxonomic knowledge. Also, only some groups or even only some species are preserved, and sometimes, they don't even preserve at all because of dissolution. Then we have biomarkers. They can be preserved in a similar time scale, so more than tens of millions of years, and they are quantitative. But they are not always source specific. There's limited knowledge on the producers of specific biomarkers. There's no direct phenotype information, also no genotype information, and it heavily depends on standards and calibration data sets. And finally, there's sedimentary ancient DNA, and we know that it doesn't preserve for as long, and currently it's limited to like more than million of years in time. But it covers all domains of life, and we can use high throughput sequencing to get really large data sets. Also, sequencing data has the potential to link specifically to certain taxa, also to certain traits. And we can use it potentially in a quantitative way if we look, for example, in molecule numbers. However, there's no direct phenotype information. Then we have problems with gaps and mistakes in reference databases, so we have sometimes misclassifications. There are bacteria and archaea and also fungi in sediments that can completely overprint eukaryote signals. And of course, there are also risks with chimera formation and contamination, and also risks of biases in extraction methods. So if if you're comparing the results of different labs that use different extraction methods, you might come to different conclusions. However, we are really happy about sediments, and we are having different ways of getting to these sediments. So for example, if we target Holocene or younger sediments, we can use gravity coring, and by that we are typically getting sediment cores of like 3 meters to 8 or 9 meters length. And if we want to go further back in time, we need more sophisticated methods to reach the depths where these sediments are buried. So for that, we could go on a drill ship, for example, in the IODP program. So you can already see from these pictures that a much larger collaborative endeavor is needed here. And here I have a picture of the drilling vessel JOIDES Resolution, where I've been on in 2023. And it has this huge drill tower, and pipe after pipe is lowered down through the water column and into the sediments, hundreds of meters deep into these sediments to get out these sediment cores. And here the cores are typically 10 meters long, but one core after another comes up, and the operation goes on 24 hours. So the ship never really sleeps. So typically when we subsample samples or like a sediment cores that have been taken up by gravity coring, we take the cores home, and then we split them open in a lab under controlled conditions and subsample them there. But on a drill ship such as IODP, this is not possible. So there, the 10-meter long cores were chopped up in 1.5 meter long sections, and I immediately took samples at the end of freshly cut sections. However, with this method, you might miss an important interglacial because you're just randomly sampling. And therefore, additionally, we took also samples on freshly split cores. And what you can see here is that we are always wearing these protective clothes because we don't want to introduce any contamination from ourselves or our microbiomes on the samples, because this could also, again, override our signals that we have in these low biomass environments. And on top of that, for IODP, we are drilling. So for that, we added a perfluorocarbon tracer into the drill fluids to monitor contamination, because it uses seawater, and that could also completely contaminate our cores. So next to a DNA sample, we also took a sample to look for this tracer and via gas chromatography. Once we have all these samples collected, we bring them into a dedicated sedimentary ancient DNA laboratory. And here I just wanted to highlight that we just got a brand new ancient DNA lab at GEOS, and we're now starting to process all these samples there. Okay, now I want to finally get into our data and want to start with my first of 2 case studies. And here I want to show biodiversity and functional trade responses to a Holocene climate in the marine Arctic. Again, I want to bring you back to this really nice picture by Cristiano Sade, and I want to highlight all the different body types that we see. We see some species here that are phototrophic, some of them are heterotrophic or parasitic. So it is really hard to just take one group and say this is phytoplankton, and the other group this is zooplankton. It just doesn't capture all the variability in there. And for that, I wanted to look more into traits, because they capture more of the attributes of the functional attributes of the species. So I looked into resource acquisition, growth, and survival. And on top of that, I looked into traits that are morphological, behavioral, physiological, or associated history of the species. And it is important because traits can define the fitness of a species in a given environment. So we have a certain diversity in species composition, but environmental conditions, they can act as a filter on certain traits, and therefore also on the diversity and the composition. But other traits, so-called effect traits, they really affect ecosystem functions and thereby also ecosystem services that are so important for us humans. On top of that, effect traits can also give a feedback on environmental conditions. And I was working on a Holocene sediment core from the Upernavik region in northwest Greenland. And this core was taken at almost 1 kilometer of water depth, and it's more than 7 meters long. And over these 7 meters, we took 38 samples in triplicates to process them for ancient DNA. And here I want to show you quickly the age model, but I just want to highlight that the core encompasses the last approximately 9,000 years and therefore the Holocene thermal maximum and also the neoglacial period. The method I'm using here is called DNA metabarcoding, and for that I extract DNA first and then we use PCR. So now after COVID, I guess everybody is kind of familiar with PCR. And for that I was using two different markers. So instead of testing if somebody has COVID or not, I'm looking into or amplifying the 18S gene, that is a broad eukaryote marker, and a marker that is specific for diatoms based on the RBCL. And after that, after the PCRs, we sequence all of that, and the sequences we retrieve are quality filtered, and then we compare them with a reference database for taxonomic assignments. And here I went one step further, and after taxonomic assignments, I looked into trade databases and the literature to collect all kinds of traits for especially the protists. And after that, I used clustering to cluster the protist data into functional groups. And here are now the results, but I got about 8 different functional groups, and I just want to highlight 3. First of all, I want to highlight the parasites and the saprotrophs, because this shows that the highest diversity is actually in groups that are non-fossilizing. And on top of that, we see, for example, for phototrophic floaters, a group that contains only diatoms here, that about like 4,200 years ago, there's a shift in diversity. And for parasites, the same. The diversity is declining, and then it's at a stable mean and just variable over time. So again, at around 4,200 years, where we have the boundary from the mid to the late Holocene, and we think this is associated with the neoglacial cooling. Furthermore, we used a proxy for temperature and a proxy for sea ice to correlate our diversity with to correlate our diversities with, and we saw that phototrophic floaters are highly negatively correlated with temperature, and also the saprotrophs are negatively correlated with temperature. So this means we actually have higher diversity of these groups under colder conditions. For multicellular groups, I disentangled the taxa into groups by habitat. So we have the pelagic fauna and we have the benthic infauna, so the species that bury themselves into the sediment, and also the benthic epifauna that lives on top of the sediments. And again, we see, for example, for pelagic fauna that there's a shift in diversity at this mid to late Holocene boundary. And here again, there's a negative correlation with temperature. Overall, there was a higher diversity found for benthic epifauna than for pelagic fauna, and this is probably because the benthic community is living on the seafloor, and it is quicker the DNA is quicker incorporated into the sediments compared to the pelagic fauna that where the DNA is in the water column and might get recycled. What we see here for the benthic infauna is, however, no correlation with temperature, but a negative correlation to sea ice. And interestingly here was that when we looked into or compare the peaks of the higher diversity with the with the reconstructions for paleoproductivity, we see that that the peak corresponds also to this peak in higher productivity as estimated by diatom microfossils. For benthic epifauna, we see that that there is a quasi-cyclic periodicity in the data. So this is also an interesting pattern that we see. Overall, we see that the taxa that are positively or negatively associated either with temperature or sea ice are taking over very important ecosystem functions. So for example, we have primary production, carbon export, trophic transfer of energy, remineralization, nutrient recycling, and bioturbation. So this has an important component towards what will happen in the future under climate change. How will this affect ecosystem services such as provisioning, so for example, seafood, or regulation, for example, of climate and the atmosphere via the carbon pump, biological regulation and pathogen control, and life cycle maintenance. And now I'm quickly going towards the second study I am on, and this is my involvement in the IODP. So I have a 1 million year record of Arctic climate change. And we were 27 scientists from all across the world, and we again went to Northwest Greenland. So overall, DNA can be preserved over up to 2 million years of time, and the oldest DNA that we found so far was from marine sediments, was from Antarctica, dated to 1 million years ago. But most of the samples we have are actually falling into the Holocene and the late Pleistocene, so there is a relatively big gap in the middle and early Pleistocene. And with my research, I want to address this gap. And I'm tackling the questions how Arctic marine ecosystems have reacted to past climate and cryosphere dynamics. There's a huge challenge of retrieving sedimentary ancient DNA because the deeper you go into the sediments, the less DNA is there. And of course, most of that DNA is actually from bacteria. So eukaryotes are just very small fraction. And over time, the fragments get smaller and post-mortem damage increases because repair mechanisms don't work anymore. We get to use a little trick to enrich for eukaryote DNA, and we use an approach called hybridization capture. And this is a project of my postdoc, Jérémy Quentin, who developed an assay to enrich for barcode markers, and this contains more than 11,000 species in 58 phyla. So what we found was that overall, when we extracted DNA from a very small subset of only 42 samples and duplicates, that DNA is preserved down to 237 meters below the seafloor, which is already really great. However, there's very, very little DNA, and especially in the deeper part of the core, it is very, very low. We took 10 libraries or 10 samples that look promising for this capture enrichment and sequence it on a NovaSeq. And now I would just want to show you the microeukaryote diversity, and I put it into context with a delta 18O stack, and in red you can see the interglacials that are highlighted there. So overall, there's relatively high richness in both glacials and interglacials, which was surprising, and the highest richness is found in the Holocene. However, there's also relatively high richness between 500 and about 600,000 years ago. And now I want to show you the most abundant microeukaryote phyla, and maybe you know already diatoms. They were really abundant in this data set. Foraminifera and haptophytes are also quite abundant, and those are fossilizing groups. However, cercozoans, chlorophytes, ciliates, or Euglenozoa are not really fossilizing taxa. So we have a very nice range of trophic modes in this data set, and we have a very nice signal of diatoms. Even we did not find any diatom microfossils in this core. But we don't just have a high abundance of diatoms, we also have a relatively high diversity. We found 32 species in 14 different families. And I quickly want to highlight the most abundant ones on genus level. So we have genera that are typical for this region, such as Chetoceros and Talostyra. But we also have genera that contain species of cold water and that are thriving in cold water and sea ice, such as Spectorozyra, Fragilariopsis, Nitzschia, and Porozyra. And additionally, we find Pseudonychia, and this was mostly assigned to a domoic acid producing Pseudonychia obtusa, and it falls within the marine isotope stage 13. So it could be a warning signal that under a future warming Arctic, there might be more of these potential toxin producers in the Arctic. As an outlook, because this is all relatively fresh work and we just got started, and there's so much more that we want to do. We currently are working on refining our extraction methods, and then we will do deep shotgun sequencing to characterize the taxonomic and also the functional profiles in these cores. And we will use that information combined also with more hybridization capture to look into what are the main players of the nitrogen cycle and the carbon cycle over these glacial interglacial cycles, and how does the nitrogen cycle affect the primary producers and secondary consumers, and what that means for the biological carbon pump. Finally, we want to look into the analog potential and use past interglacials to model future biodiversity. But the IDP samples are not the only things we are working on. We are currently compiling shotgun sequencing in a Greenland and an Arctic perspective to create natural baselines for biodiversity over time, and to look into marine ecosystem resilience and the sensitivity to climate change. So the take-home messages I have for you today are that DNA really has a potential to understand the long-term responses of a vast diversity of species, even beyond just simple taxonomy, and especially for non-fossilizing groups, or in the absence of microfossils, as here was the case with diatoms. And this really highlights the potential for modeling Arctic biodiversity under future climate change. Also, we had very recent advances with methods and sequencing technologies, and this really allows us to probe previously unobserved time intervals, of course, only for DNA. And I think this will open a window to understand the role of climate in shaping evolution, extinction, and adaptation. And with this, I want to thank you.
So are we going to move to the next stage with the questions now?
Yeah.
Yes.
So,
Danny, I will need some help from you because you have been with CP for much longer time, and there is a question that we got regarding the basically what has been done over 20 years to promote early career researchers, right? So I could answer some part of this question, but I will need some help with this.
But yeah, this is a question for the speakers. So what do you see as the most significant shifts in polar research methodology over the last 2 decades?
But I think that this is actually a question to us.
Yeah, but maybe the speakers can, I would prefer the speakers to speak about that, yes? Because,
yes, what they think. That's great, yeah.
Heike or Peter?
I can say a few things. I think both our talks show the shift from descriptive research to quantitative research, right? That is where I think the past 20 years have been particularly instrumental, and also bringing different disciplines together that were previously doing their own thing separately. I think that is where not a disciplinary journal or a disciplinary topic, but like a theme topic, which I see climate of the past is what brings all these fields together, is what has happened in the past 20 years. And you see that because papers in climate of the past have lots of multi-author contributions, and that shows that you need different disciplines to bring together to come to the next level of answering questions. And we are two maybe senior scientists. I don't know how it is with Heike, but in my lab, it's the early career researchers that do the actual work. And I hope my presentation ventilated that a little bit by showing who actually did the work in every plot that I showed. And we cannot stress more that we will be nothing without our task of training the next generation of scientists in universities or institutes. So that will be my answer to this.
Eike, you want to add something?
I think actually that especially with biomolecular work like biomarkers and also DNA this has really made a shift and let us explore new ways of either identifying the new ways of reconstructing past environments, but also understanding the responses, for example, of ecosystems to climate. And I totally agree that the new generation is very important and does most of the work. So
excellent, thank you. The CP also ran a special issue focused on the past glaciations recently, and I think we got a lot of publications from young scientists in there.
Okay, so the next question is from Erin McKeeman to Eike. It's you mentioned that the preservation of the DNA can also be challenging. Does this also change with time? And if so, is there a way to assess the impact of changing preservation on the reconstructions you have made?
So preservation is a huge part of the work that we are doing currently, like identifying what helps the DNA to be better preserved over time. And there are so many different factors. So of course, age is an important factor, but this is not the only, not probably the most important one. So we also know that the DNA mineral interactions are very important. So clays, for example, they are really strongly binding DNA, but currently our DNA extraction methods are not releasing a lot of that DNA. So as you see, the DNA concentrations were very low that I had, but we know that it's only like a fraction of we calculated that between 14 to 21% of the DNA has only been released. And that's why we currently work more on identifying which protocol is better suited to release more of that DNA. And on top of that, you, especially with IODP, where you have these long cores, hundreds of meters deep into the seafloor, you also have the effect of temperature. So we see that even over the first 120 meters, we have an increase of about 5 degrees Celsius. And this is, temperature has a huge effect on DNA preservation, but it's very, very hard to disentangle it, and we're trying our best. But currently, I think in my data set, it's obvious that the clay fraction has the most impact, I think, on the preservation.
Mm-hmm.
So following up what Eike was saying, what about the preservation of the biomarkers for your study, Peter?
Yeah, it's the same. Obviously, in the Southern Ocean sediments, preservation of organic matter, whether it's biomarkers or polynomorphs, is extremely challenging. Most of the gaps we have, for instance, in our surface sediment sample record of dinoflagellates is caused by this vast, deep ocean that is completely oxidized, and all the organics are gone, or at least gone to the like they are so degraded that you cannot really do much with it anymore. That's a challenge, and it's also a signal because if we go back to the Eocene, for instance, we have a lot of organic matter preservation, but it also tells you something about the ventilation capacity of the Southern Ocean in that time. So it's a challenge, but it's also a factor. But yeah, all the preservation potential of the dinoflagellates and the biomarkers also increase with increasing clay content. Usually, it's also usually clay bound. We are just we have some methods, quite destructive methods to get them to let go of the clay, but those will also destroy the DNA, so it's not much use to hike, unfortunately.
Irina?
Yes, I have a couple of questions if possible. Do we have
Yes.
time?
Yes, we have.
Peter, yeah, I'm kind of wondering, I worked with the tectonic processes and I'm interested in what kind of reconstructions do you use in your models for the
Oh yeah,
past?
so that could have been a whole presentation on itself. I worked a lot with tectonophysicists, and in parallel, they were making these paleogeographic reconstructions using a paleomag reference frame.
Uh-huh.
And that basically reconstructs more directly and accurately the position of the spin axis relative to your site or your continent of interest.
Uh-huh.
And for paleoclimatologists, that is really important, of course. So, I mean, your paleo latitude or your geographic position of your continent or site is really relative to the spin axis because that determines your latitude and that determines for a large part your local climate, right? And so for us, it was really important to move also the modeling community, which wasn't there yet, towards making paleogeographic reconstructions in a paleomag reference frame rather than a moving hotspot or whatever there was, because it made a huge difference. So we have done this Eocene simulation under these two geographic boundary conditions. So the geography was the same and the relative position of continents relative to another was the same. But then you basically change the position of the spin axis of the Earth in that tectonic circuit. And it means for the Eocene that Antarctica was much further north on the Pacific side and south on the Atlantic side. So it makes a huge difference for the position of the ACC relative to Antarctica and relative to your site, the ideal flow path of the ACC over of any ocean flow. So it makes a big difference. Otherwise, you are comparing apples and oranges if you compare your proxy data with your modeling results.
Yeah, absolutely. What kind of sensitivity tests did you run with different reconstructions?
Sorry, what is it?
Sorry, I will move a bit closer. The sensitivity test, did you perform some just to understand how much it influences the results?
Yeah, so we basically run the same model under these two geographic boundary conditions.
Okay. Yeah, two.
And then see the result, see the differences. And that's work by Bijl. I think there's also two papers in Climate of the Past that demonstrates that. And so we added to that also the temperature proxy and compared the latitudinal temperature gradient from the two model simulations to the proxy data and see whether it made a difference. And yeah, so it didn't explain everything, but it was it's still important.
We will never have a definite answer.
No.
That's our job, to look for it.
Exactly.
Okay. I have also a couple of questions for you, if you are ready to answer. What can your climate proxies tell about the timing of when the breakup happened, really, of the Gondwana? Like, can you establish the timing of different parts of Gondwana? I mean, I saw some periods that you showed, but can you be more precise with this in certain parts?
Yes, we are a bit limited by the geographical spread of the sites. But for instance, what I didn't really show is that we also reconstructed sort of the first small opening of the Tasmanian Gateway. That was around 50 million years ago when things also start cooling. It's all very coincidental at these places where you see things changing in temperature. That's also when the gateways in the southern hemisphere did things. But this is right at the end of the early Eocene. We see the first opening of the Tasmanian Gateway because we see these endemic dinoflagellate cysts, these blue ones that I showed in this map. We see them all of a sudden appearing in the Australian Antarctic Gulf, so it shows sort of a countercurrent through flow. And then also what was in the modeling paper by Isabel Sauermilch on the deepening of the gateways. We also compared that to the proxy data in detail across the Eocene and Oligocene transition, and it also made sense that exactly at the EOT, sort of the subpolar gyre was getting less prominent and was basically overridden by the earlier the proto-ACC.
Thank you. I was a bit puzzled, Heike, maybe you can join the discussion a bit, maybe both of you. Like sea ice, the discussion that we had about sea ice. Is it also confirmed somehow by recent observations that the sea ice extent is shrinking the biodiversity? Like when it's, because it is shrinking really fast right now in the Arctic, right? So can we say something about this based on the observations?
Right now, I can only tell that from what I'm seeing in the past is that in colder periods we have more diversity, and the Arctic is highly specialized. So you have you have these often endemic species there that are also adapted to this, not just like cold climate, but also to these long stretches of polar night and polar sun. So they are quite specialized to that, and a lot of other species from more temperate regions, they might not be so adapted to that. So if we think about climate warming and shrinking sea ice and other species being possible to migrate, there might be a limit to where other species can migrate, and like the survival of this polar night and polar day. So I guess that I guess that in the future, with like less and less sea ice, the diversity will maybe first increase because more species are coming, but also at some point that there will be a decrease again because endemics might get lost with less and less sea ice.
Wow, interesting. Denny, do you have some questions?
Oh, you are muted.
No, I leave it to you.
Oi!
Yeah, you're the specialist.
I am not at all. I am acting here as a complete dummy. I'm sorry, because that's not my subject. I'm dealing with the ice sheet and glacier modeling.
Yes, but still.
But I am super interested in this. Okay, okay. I was puzzled by one thing that you were talking about, like that during 500 to 600,000 years ago, this period, there was a very, I don't know, like extensive diatom richness. I don't know how to ask it properly because that's really not my subject. Could you talk a bit more about this, Heike?
Yeah, there's this especially there's this one peak that is the it's about 500, maybe 50,000, dated to about 550,000 years, but we actually think because the age model right now is super crude, I just interpolated between paleomagnetic tie points, so we don't really accommodate for variable sedimentation rates between glacial and interglacial. So I guess that this like peak actually falls into the marine isotope stage 13, because right now it's a little bit outside, but I think because of the composition, it looks much more like an interglacial compared to a glacial. And that's I think that is super interesting because from the diatom microfossil assemblage we get nothing. There's nothing in this core. It's all dissolved. So that's why it is so important to look also maybe if like possible into DNA, because otherwise there's nothing we can get if diatoms are dissolved. Yeah.
It is fascinating because it was even before MIS 11. Which was the longest and the warmest, let's say. I wouldn't know. Really?
Also, we have relatively low diversity in marine isotope stage 5. So the last interglacial, I guess, with a better age control. like we might say a bit more in which time interval this falls. It might not be like hitting the Eemian. It might be a bit later and or a bit earlier. So yeah.
Yeah. But how certain are we that we did have this, let's say, explosion of diatoms during 500 to 600 or whatever the period was? Is there any certainty that we can put on it, really, based on the dating methods?
So based on the paleogenetic type points, it is in this like interval. Like there's it is definitely there. We have diatoms there, and quite a lot of them, and quite a big diversity. But so like depending on the time scales we're talking about and the Arctic, but yeah. I would say we can trust that there is a signal, but we cannot trust that it's exactly at like this time point. Yeah.
Irina, we have two questions in the Q&A box.
Yeah. Okay. Yeah. Yes.
So for Don Slibos, a general question for both speakers. Do you perhaps have insights into phylogenetic relationships of past and present proxies, and how these relationships relate to differences in their survivability to contemporary sea surface temperatures or sea ice conditions?
That's a mouthful. No, I think I understand the question. I mean, if I speak for dinoflagellate cysts, that's a whole subfield trying to connect cyst to its producer, to its living cell.
Yeah.
They have separate taxonomies, which also illustrates how difficult this is. Otherwise, you would just name it the cyst of a motile cell species. But I think in general, also ancient DNA could help there a little bit. And I think that is also still a little bit underexplored. I think also because ancient DNA takes a lot of time, and there are more like bigger fishes to fry than my simple dinoflagellate. But I know a few examples where this was done. Unfortunately, that is not all of that is published. But to compare assemblage changes in fossils to assemblage changes in or like abundance changes in DNA is not as easy as it may sound from the beginning. So, but yeah, I don't know if you want to say anything about it, Heike.
Yeah, actually, I can agree, because I have a paper actually about that in review, where I'm trying to connect the DNA with the microfossil records of diatoms and dinoflagellates, and this, like especially the dinoflagellates, they were a real pain to combine our knowledge from the cysts with the taxonomy of the motile stages, and then also to the DNA, and we actually find that this is completely complementary. So most of the cyst taxa we actually didn't find in the DNA, only a few of them. But on the other side, we have a lot of different dinoflagellates that we do not find in the cyst taxa, because they're not producing these cysts. A lot of them are parasitic, so Syndynialis, for example, but also a lot of Gymnodynialis. And for diatoms, it looks a little bit better. And I think it might be actually like quite relatable, but right now we have, especially for the Arctic, a huge database gap. So even if we have the DNA and we have the microfossils, like we cannot bring them really together, because it's maybe an unknown in our record, and because we don't have a reference sequence for them. And those are major players, also indicators of sea ice, for example, but it's hard to culture them and to get these references. So it's hard to bring them together, but the community is working on it. It's just not really easy. And right now we're looking mostly just into what is there and what is absent, and how can we bring them together. And we haven't even really like started into like phylogeny, but yeah, this would be the next step, definitely.
But I
Great.
think in any microfossil record that I've seen for polar regions, whether it's from DNA or from microfossils or whatever, it is so clear how strong these microfossil communities change to environmental conditions. So related to the second part of your question, I don't think it should be a really fruitless aim to try and keep the ecosystem alive when conditions change, because it's just not their niche. And there's not only temperature, there's all kinds of, it's the combination of the whole environmental conditions, of course, that make these species live where they live. And so if there's one thing we learned from these assemblages in the past, is that the ecosystem will change if conditions change, and rapidly. So that is maybe it's an open door, but I think yeah, it is what we see, and particularly in polar regions.
Yeah. So just to a last question for both of you before closing the session. In a perspective of 20 years of polar studies, what do you think are the promising directions of paleo studies in the Arctic and Antarctic regions?
Maybe we can also join this answer.
So I prefer the speakers.
Yes, for sure, for sure.
I could start a little bit.
Yes.
I think that we need to do a lot more multi-proxy studies, get like various pieces of the puzzle together, and including more ancient DNA also. Of course, I have to promote myself, but this gives a whole new perspective on the past, and I think collaboration across disciplines is very important. Also to bridge these gaps between DNA and microfossil records. So yeah.
Peter?
Yeah, and I think a lot of the proxies that are developed by people in their core discipline are now ready to be applied by people that maybe not are super specialists, right? I speak for myself. I'm not an organic geochemist, but I'm applying all these biomarker proxies as a paleoceanographer. And so I think the maturity of some of these proxies are such that a much wider part of the community can actually start to use them. And I think that is promising in a way, is that it also brings disciplines together in another way, not only by co-authorships in papers, but also just having the community applying these things and bringing it together there. And that's a promising direction. I think the most important challenge, if I can add, is that we have to keep the infrastructure going that allows us to go to these polar regions, because it's not only IUDP, but on all kinds of fronts. Infrastructure in polar regions is very, very heavily under threat, and we should really strive hard to convince government to keep things accessible there.
Yeah, we are facing very special context currently. Yeah.
Exactly.
Yeah. So thank you again to Peter and Heikki for their stimulating presentations and replies, and thank you all for attending and for these interesting questions. The next webinar is scheduled next month, Wednesday, February 11, and it will focus on paleomonsoon variability. So we changed, and it will feature two exciting presentations by Wu Yongyu and A Cheng. So the registration will open soon on CP webpage. So again, thank you very much for this very stimulating webinar, and yeah, see you very soon.
Thank you.
Thank you. Bye.
Bye.